Hello, and welcome back to Human Evolution. Today we want to talk about the second step of DNA to proteins, and that is RNA to proteins. So we're talking about translation. So remember, the central dogma of molecular biology is that idea of taking DNA, which is the instructions, transcribing it into RNA, which is the messenger, that goes from the nucleus to the cytoplasm, where that mRNA is translated into a protein. And so today we really want to focus on how that translation thing happens. But for starters, I just want to ask, what are some of the functions of proteins? So in this case, which is not a function of proteins? And the reason I'm asking you this is because it seems odd if you just have to eat protein to build muscle and stuff like that. Why would we care so much about proteins? So, which of the following isn't a function of proteins? Facilitating anabolic and catabolic processes, building connective tissues, fighting antigens like bacteria and viruses, transmitting messages from nerves to receiving cells. All of these or none of these? Well, it turns out all of these are functions of proteins and so many more. Proteins do like everything, and we will spend a lot of time talking about what proteins actually do. But rest assured, they do lots of things, and so we really want to make lots of them. So how do you do that? The first thing is you have to put amino acids, which are the building blocks of proteins, together in a chain. So when we talk about building DNA, we chain together nucleic acids. When we build proteins, we chain together amino acids, and each amino acid has a central carbon, a carboxylic acid group, an amine group, and then some sort of unique functional group. There are lots more amino acids than there are nucleic acids, but they're both organic acids, and they both make up these two really important macromolecules. All proteins are made up of a combination of 20 amino acids. There are only 20, right? There's only 20. 9 of them for humans are essential, and 11 of them are non-essential. And you may have heard this, right, when people talk about, are you getting your essential amino acids? Well, what that means is that 9 of those 20 amino acids can't actually be synthesized by the human body, and so we have to eat them. It is species dependent as to which ones are essential and non-essential, so this 9-11 split is for humans. Okay, and I've put in here a list you can see of which ones we have to eat and which ones we can actually make ourselves. Okay, so let's talk a little bit about translation itself. Translation is more complicated than transcription. Translation has 3 steps, just like every other process, which is initiation, elongation, and termination, but there's a little bit more going on. So what we're going to start with is initiation. The very first thing that has to happen, actually, before any of this happens, is that the mRNA has to exit the nucleus. We know that it needs that 5' cap, that poly A tail, right, and it has to go through the nuclear envelope because transcription occurred where the DNA is in the nucleus, and translation has to occur where the ribosomes are in the cytoplasm. So the very first thing that's going to happen once the mRNA gets out into the cytoplasm is the bottom part of a ribosome, and you can see that here. This is the small ribosomal subunit, this is the bottom part, is going to bind to the mRNA strand at a special location. And that special location is marked by the sequence AUG. We will always be looking for the sequence AUG. This is how the ribosome knows where to go. The ribosome is going to read the mRNA from 5' to 3'. So when we were talking about transcribing, right, we were actually following that 3' to 5' direction on the coding strand, right, and then we were growing on the 3' end of the mRNA. Okay, but in this case, that mRNA, remember I always say things are read 5' to 3'. Right? Replication and transcription are occurring from, when you look at the template, right, you're going from that 3' to 5'. But in translation, we're reading 5' to 3'. So this is the one that's backwards from the ones we've done before. Okay, so we're going to look for that AUG. We're going to orient the ribosome so it will travel in the 5' to 3' direction. Now, once it's set up, there's going to be an initiator RNA piece. It's a type of RNA called tRNA. It's called transfer RNA. I'm going to talk about it more in just a minute. But just understand that this whole process, translation, requires 3 different kinds of RNA. The messenger RNA that was made in transcription, the tRNA, which was actually also made during transcription, but it was in those sections that were cut out during the pre-mRNA processing. Okay, then we've got the small and large ribosomal subunits which will be involved. Those are both made of rRNA, which is ribosomal RNA. Now there's some proteins involved in forming the ribosome, so you get this complex of rRNA and proteins. But we've got 3 RNAs that we're working with. The mRNA, the message, the tRNA, the sort of workhorse, and then the rRNA, the ribosome, which is the actual little factory. Okay, so a tRNA is going to bind to that special sequence here in the mRNA, AUG. Our ribosome is also binding to it. Once these 2 things are in place, the large ribosomal subunit is going to seat itself directly in the center of that AUG, right? So it's going to surround this tRNA. It's going to hook on to the small ribosomal subunit, and you get this cute little hamburger setup. Okay. Once you have this setup, then elongation can occur. Okay, so just to reiterate, you've got an mRNA sequence, it leaves the nucleus, a small ribosomal subunit finds the AUG in the sequence, a tRNA also finds the AUG sequence. They both latch on. Once they're both seated, the large ribosomal subunit will complete the ribosome, and we're ready to go. Okay, now inside the ribosome there are 3 slots where tRNA can be. Okay, now what is tRNA's job? tRNA carries those amino acids. So you remember I told you that tRNA is the workhorse. It is going to carry around an amino acid. So the tRNA will bring whatever amino acid it happens to be carrying into the ribosome, and then the ribosome is going to be able to remove that amino acid, hook it to an amino acid chain that it's forming, right, and then eject the tRNA out of the back of the ribosome. So what happens is the tRNA, once we start elongation, we've got that first tRNA sitting in what's called the P site. Okay, then we have these 2 open sites on either side, because remember I told you there were 3 slots in the ribosomes. There are 3 slots in the ribosomes. Okay, so we've got that initial one. Now a new one carrying a different amino acid is going to pop into the A site, which is the acceptance site. Okay, and now we are ready to begin building the chain. So what's going to happen is that the ribosome is going to slide down the mRNA. So the mRNA is going to kind of stay what it's doing. The ribosome is going to slide down it, and then a new piece of the mRNA will be open. So as this slides, you're going to get a new piece that's open. All right, and then a new polypeptide is going to come in, and when it does, it's going to, a new tRNA is going to come in, not polypeptide, a new tRNA is going to come into the A site. The one that was in the A site is going to move into the P site because they stay attached. The tRNA attaches to the mRNA and it stays that way. And so as the ribosome moves, these guys stay where they are. And so the slots slide around the tRNA. So as this guy comes into the acceptance site, then the ribosome is going to move. This guy will end up in this site. This guy will end up in this site, right? And then when the empty one ends up in the exit site, the E site, the exit site, it's going to get kicked out. Okay, now I'm going to go over this one more time, but before I do that, I want to talk a little bit about how do we know which amino acid is next. Okay, so here's the thing. Yeah, DNA is red, right? in little sections of 3, right? So let's say you've got this DNA and you've got a sequence on the template strand that's CCA, okay? Our mRNA would be GGU, and this would reflect the coding strand, which would be GGT. Okay, so in the DNA, you'd have a coding strand that's GGT. We're going to use the template strand to attract the mRNA to build the right mRNA, which is transcription. It's GGU. Now, we're going to read this as all the mRNA is read in groups of 3. Each sequence, group of 3, is called a codon. So if you've got your DNA sequence over here, let's say that I have my template strand is TAC, CGA, GAT, TCA. Well, my mRNA sequence, I can break that up into little words, right? Codons are DNA, RNA words, right? So my first word or codon will be AUG. My second codon, right, will be GCU. My third codon will be CUA, and my fourth codon will be AGU. So I'm always going to read, and the ribosome will move 3 nucleotides at a time because it wants to get the whole word open. All right, so then I can look at what my codon is, and I can know what the amino acid is, and I'm going to show you how we do that in just a second. But in this case, the amino acid is going to be methionine, alanine, leucine, and serine. Okay. Now, one thing to note is that, remember, I told you the mRNA and the tRNA bind. Well, RNA has the same binding rules that DNA has. It's always complementary, and it's always CG and UA, right? Whereas in DNA it was TA, in RNA it's going to be UA. So on the tRNA there is an anticodon, and I'll talk about that more in just a minute, the anticodon in tRNA. But the tRNA is going to bind complementarily, right, to the mRNA. Okay. Now, how do we know which amino acid the codon codes for, right? If you think about, there are 4 nucleotides, right? And you start doing all the combinations, there are 64 possible codons, right? You could have UUU, you could have CAA, you could have AAG, you could have GCC, whatever, right? But there are only 20 amino acids. So what this means is that multiple codons can code for the same amino acid. Now, no codon can code for more than one amino acid, but many amino acids can code for many codons can code for one single amino acid, right? So if you look at leucine, there are actually 6 different codons that give you leucine. So if you get a mistake in the DNA, for instance, and you have CUC, but there's a mutation and you get CUA, it doesn't matter because they both code for leucine, and that's very cool, okay? We'll talk about that in a second. So the other thing that you need to know is that there are 4 special codons. The first one, AUG, I've already mentioned, and that is the start codon, right? AUG is methionine, or start. AUG only codes for methionine, and methionine only has one codon, AUG. Okay, that's the start codon. It's very important, right? Now, there are 3 codons that code for nothing. AUU, UAG, and UGA, they're just stop signals. There's no tRNA that matches them. There's no amino acid that matches them. So in reality, there are 61 codons that code for amino acids. 3 of the possible codons tell the ribosome that it's time to stop, and we'll talk about that when we talk about termination. Okay, so here's your next question. The genetic code is often called degenerate. What does that mean? The genetic code is the same in all organisms. More than one codon can represent one amino acid. Some codons can cause translation to stop, and codons can change through mutation. Well, when we talk about the genetic code as being degenerate, we mean that more than one codon can represent one amino acid. So I'm going to go back to this slide for just a second. This is what I was telling you. This is a redundancy system. The sequence of amino acids is really, really important, and I'm going to talk about that more in just a second. But if you change the sequence of amino acids, you change the protein. So if you get a mistake in the DNA, you don't want to change the amino acid sequence. And so it's very helpful, it's protective to have multiple codons coding for one amino acid just in case you make a mistake. And what you'll notice is many of them have the first two nucleotides are the same, and then you have some variability in the third nucleotide. That third nucleotide is called the wobble position, and because of the way that the tRNA binds, sometimes you can get the wrong tRNA to bind with a particular codon, not coding, codon, and that third one may actually not be the right one. It doesn't bind as tightly, and so you can sort of get a little bit of forcing going on there. And so it's not uncommon for you to have a situation where that third one may not be correct. So to have all of these guys code for proline, right, means that you don't have to worry about this weird wobble position that has a tendency to make mistakes. Okay, and we'll talk more about this when we talk about mutations, but this is one of the things that protects you from extra mutations, is having this degenerate code for codons. Okay, so let's pick up where we were before, now that we understand what codons are. So just to review, the mRNA is read in three-letter words called codons, so 3 nucleotides at a time. The ribosome attaches to the mRNA. It has 3 sites in it, where tRNA carries amino acids, they're like amino acid baskets, can slide in, and then the amino acids can be put together in a chain by the ribosome as it goes across, like down the mRNA. So 5' to 3', we're going to latch on to that start codon. Okay, we're going to latch on to the start codon, and then the small and large ribosomal subunits form the big ribosome. Then, right, we've got the one. We're going to start getting tRNAs to come into the acceptance site. The ribosome is going to move 3 nucleotides down. The tRNA is bound to the mRNA until it's released, so it doesn't move from the codon it's on, right? So when this comes in, codon recognition, then we're going to have a bond form as the ribosome is moving. And when the ribosome moves, the tRNA, this is called translocation, the tRNA that is in the P site will move to the E site, which is the exit site. The one that was in the acceptance site, okay, so this one is the one that was in the P site. The one that's in the acceptance site will move into the P site, okay? Now, once that happens, as the ribosome moves, you now have an open acceptance site. A new codon is visible. You've got the old tRNA, which has the entire chain on it, because the chain is popped onto the top of this guy. So you see this guy came in with the square. This circle was bound to the top of the little square there. These are 2 different amino acids. So when this moved, it had the entire peptide chain, polypeptide. Okay. The original tRNA was kicked out the E site, and now we're ready to start again because the A site is open. So we get a new guy come in. The polypeptide, the growing polypeptide, shifts over to that tRNA that's in the acceptance site. The peptide bond is formed. The peptide bond is what holds the amino acids together. And then the ribosome moves. The one that's in the P site, the tRNA that's in the P site, gets kicked out. The tRNA that is in the A site moves to the P site. And now the A site is open again. You get another tRNA in that has a different amino acid that matches the new codon that's open, and you keep going until you hit the termination point. The termination point is a stop codon, okay? And so there's no tRNA that's going to bind here. This causes the ribosome to destabilize. It disconnects. It releases both the polypeptide and the tRNA, and then the ribosome can go off and do something else, like translate another mRNA, whatever, it's free to go, and now you have your little polypeptide. So when we put it all together, right, we're going to have the start codon, that AUG, is going to be the indicator that we should start translation here. The ribosome will bind to it, as will a tRNA carrying methionine, right, because that AUG. Then we will start into elongation, where the ribosome will move 3 nucleotides or 1 codon over. The acceptance site is open. A tRNA that has a complementary code to that will plug into that A site. The methionine carrying tRNA will have moved into the E site after the polypeptide is formed as the ribosome moves, and then the A site is open again. We've now got a polypeptide on that second tRNA that had come into the acceptance site that's now in the P site. There are 2 amino acids on it. The tRNA that was carrying the methionine is kicked out. Okay, so now we're going to bring in the third tRNA. That third tRNA is going to pop into the acceptance site. Its anticodon will be complementary to the codon. We'll have the polypeptide bond form, so that will move over to that third new tRNA as the ribosome shifts, and the tRNA, the second one that came in, will now shift into the exit site and be kicked out, right? And the acceptance site is open again, okay? And that's the whole process. Remember, transcription occurs in the nucleus, translation occurs in the cytoplasm, okay? Now, let's talk a little bit about tRNA, just so we can understand that as well. Okay, so remember, when we're talking about tRNA, we're talking about an RNA sequence, but the difference is, remember that RNA is single-stranded, which allows it to bind to itself. And so, unlike the mRNA, which is a single strand, tRNA has bound to itself in this very interesting sort of clover kind of shape, although in reality, it looks like a little bent cluster because these loops aren't spread out like that. They actually fold over. But the important part about this tRNA is that, one, it has this little sequence here at the bottom of this loop called the anticodon, which is what we've been talking about. It's this sequence right here that will be complementary to the codon, and it will bind, right? And this is going to happen in the acceptance site. It will stay bound until it exits in the E site, and remember, the ribosome moves around it. Now, the other important site is this amino acid attachment site up here, and I've used AA to mean amino acids. We talked a little bit about the wobble site already, but this right here is the wobble site. This is that third nucleotide that can sometimes get iffy, which is why the multiple codons coding for single amino acids is a really important thing. But here's the amino acid attachment site. This is where the polypeptide bond is going to form inside the ribosome. And then this is the anticodon that attaches to the codon. Remember, it is complementary. So the anticodon and the template strand of DNA should match, right? And then the mRNA and the coding strand should match. Okay, now, when is that polypeptide actually a protein? So a polypeptide is just an amino acid chain, right? It ranges in size from a few amino acids to thousands of amino acids. But a functional protein has a little has some differences, okay? First of all, a functional protein can actually be one or more polypeptides. And if you think about hemoglobin, hemoglobin actually has 4 polypeptides that are required to make the hemoglobin protein. So that's one thing that you need to know about proteins, is that they may be one or more polypeptide chains, okay? Now, the next thing, do all proteins have to be folded to function properly? They do. That is true. All proteins must be folded properly in order to function properly. A lot of time, well, it's the folding that actually gives the protein its shape, and we know that its shape matters very much to the function of the protein. And the folding is unique dependent on the amino acid sequence. So if you change the amino acid sequence, right? It could change the folding of that protein, and that's why that wobble position is so important, because you don't want to change the amino acid sequence. Now, later on, we'll talk a little bit, when we talk about mutations, about some options for ways that you can have a mutation or a mistake and still get the same properly folded protein, okay? Now, so these are the two things that we know so far. It's got to be properly folded, and functional proteins may have more than one polypeptide. Now, the last thing that happens is what we call a post-translational modification. Different molecules can be attached to the protein that actually changes its function. Okay, and we change its function by doing what's called changing the conformation. And conformation is just the form, the way it's folded, what's in it, the way it reacts, that sort of thing. Okay, now here are 6 of the big most common modifications. There are actually more modifications than this, but these are the 6. You don't have to know each of these. Pick one and know that one. But basically, you can add a phosphate group. You can add a fat to it. You can add a lysine residue that'll actually make it the target of destruction, right? So if you don't need the protein anymore, or if you have a protein that's wrong, you can actually make sure that it'll be destroyed, right, and broken down. You can add cytosine residues to it. That's a pretty common thing. You can add an acetyl group, and the acetylation is opposite of ubiquitination, right? This is going to increase its stability, whereas this marks it for destruction. And then glycosylation is a really common modification, and it attaches a sugar. You can do one or more of these modifications. You can also do no modifications. But essentially, this is the second way that we get that large variety of proteins that we've been talking about when we talked about alternative splicing, right? So we could alternatively splice a particular gene to get several different mRNAs. Then those mRNAs can then have different post-translational modifications that will make them different proteins. And so we could take this one gene, and like in this case, we end up getting 8 different proteins from that single gene. 4 mRNAs, 8 proteins, right? And so you can see that this is a big thing. Yeah. Okay, that is it for translation. If you are still confused about translation, we are going to talk about this in class. Please come with questions. It's a more complicated process than we've talked about before. Okay. That's it. Thanks for listening, and I will see you next time.